Looks like we're going on Instagram as well.
Awesome. So StreamYard is working on getting more platforms, though. The downside is that the next one is that they just announced was Kick. So it's like, who still uses Kick, man?
Kick.
Yeah. Honestly, I found out about it through memes more than anything, to be honest. Yeah.
Interesting.
Right.
Animated Reptiles is on IG. How's it going? Omen Morphs. Thanks
What's up, guys?
for joining. Opus Pythons.
The white balance is all out of whack.
Big Sam Creatures with Casey.
Slightly better.
How y'all doing? Letting people jump in.
Yeah. What's up, guys?
So I am at the end of my trip that I've been on. I'll be flying back tonight. Morning, Brian.
Hey, Brian. Oh, Ben, I can tell you're a little worn out from the trip.
It's been a long week.
Oh.
It's been good seeing everybody, but yeah, I like being at home. So
for sure.
But yeah, it's all good.
Hey guys, thank you. We're excited about monsoon. Finally.
Yes. Finally put a pin in it.
Yes.
Owen Morph says, awesome catching you live. Yes. And actually UT live, like Utah. And I'm actually in Utah. So
yeah.
That's funny. Let's see. Submitted a ton of monsoon that I'm now waiting on. Yes.
All right.
Let me look and see, Brian, if I have some of that.
Yeah. Oh, that'd be cool if we could do like live monsoon results like the week we announce it.
Yeah. Can't remember for sure if we had some come through yesterday.
Brian, I know you sent some last week that got stuck in the mail. Did those ever come through? Hey, VJC Exotics. Didn't send pictures in for those. Okay, no worries.
It's being slow on me.
That's okay. It's looking like a sleepy day outside today too. Like honestly, all of May here in Virginia, it's just been like constant rain. Oh, okay, really? Yeah, looks like Okay, looks like we do have all of those, and there's another package being sent. Cool. Were the monsoons part of the ones that got sent in?
Yeah.
For anybody who's waiting on shed, I like shed results, especially if you're doing like fast testing. Feel free to like email them to, where's my little thing? There we go. If you want us to give you like live updates or test results, if they're already in, then we could probably do that here on the stream. Just email me your order ID, either put it in the comments or you can email it to me at the link that I just posted here. Oh, okay, they were all sent in, almost a year old at this point. Okay, awesome. So we should have.
Brian, do you know if they're in Texas or if they're in Virginia? And if they're in Texas, have you talked to Sean? That should get that figured out.
Yeah.
But yeah, thankfully we can finally run them and
I know.
Yes, have official results.
Oh, they are in Texas.
Okay, good. Yeah, then Sean should be able to help make sure those get going. No reason those should take very long as they know where the sheds are.
Ooh. Ooh. Hey, John. He's got some weird babies that popped out from his Dinker project, so have to send those sheds your way. Awesome. Oh, and apparently a Zuwadi was just sent in to Texas as well. Cool. I know we've been needing those.
Yep, and 283 Exotic says I just resent my ivory shed, got a weird result. Yeah, I think that was a all white snake, but did not test homozygous for ivory. So you're wondering if it's maybe a different kind of white snake. So,
Ooh.
so yeah, glad we'll definitely run that again and see if we can figure out what it is. Always fun stuff.
Cool. What's up, VA Morphs? Is it VA Morphs or Virginia Morphs? How do you pronounce it?
Yeah, either way, it is
Either
for
way.
Virginia. So Tyler's just real close to Christiansburg.
Nice. I think I might have met you then in it wasn't a Recticon, but it was like one of the little it was like in a side room in a hotel in Roanoke.
Oh,
Um
really?
that it was a show last fall maybe. Like itty bitty show, just one room, but it was cool.
Okay.
Somebody was like walking around their little caiman or something. It was pretty fun.
Oh.
Balls and strikes.
Good.
Yeah.
Well, yeah, with me still traveling, today will be a shorter show again. Probably end it at about 11:30 Utah time, 1:30 East Coast. So yeah, any questions y'all have, shoot them in. Obviously,
Yeah.
this week the biggest thing we have to talk about is that we can finally share the monsoon test. And the cool
Yay.
and kind of interesting part to that is that we did collaborate with people not only here, Charlie here in the U.S., but with Jurgen in Europe and Heather up in Canada. So it was lots and lots of work done by many
Mhm.
people. And as far as we can tell, the one we have now is a very good test. And we'll still keep looking. We're not going to give up until we feel like we found the actual causal mutation. We're pretty sure this one's very close, but very well may not be the actual causal mutation. So we're still working on that.
Yeah.
Snake Morphs one on IG says, "Woot woot monsoon." Yes, finally.
Yes.
Very glad to have that.
Oh my gosh.
Yeah.
Yeah. Made me so happy to be able to post this finally.
Yeah. Definitely felt like forever. I'm sure it did for many of you too. So
Yeah. I think Monarch is the other like most difficult one that we get the most questions about now. Would you say?
yeah.
Or what do you think it's maybe a different one?
Oh, and yeah, there's several paint and
Paint, yeah.
we also have stranger and mahogany and yeah,
Hmm.
there's many.
Oh yeah. Christian Cruz wants to know, are there any specific sheds that we're looking for right now?
If you still have that graphic, Kayla, you could put that up.
Oh, yeah, let me find where it's hiding.
Silbo Serpents on IG says, "Why are you guys releasing stuff that's not 100% accurate?" We don't have anything that's 100% accurate. We don't advertise anything as being 100%. Our line has always been over 99% accurate.
Mhm.
And the monsoon test, we have one individual that for sure does not test correctly. It
Mhm.
is visually looks like it is not a monsoon. So it does not visually look like a monsoon, but it tests homozygous. And then we have one animal that we did not expect to have monsoon in its ancestry. So we expected it to be negative, but it tested het. So that's two out of, we've probably run at least 350 known samples. So that's, well over the 99% accuracy. So yeah, we definitely will never tell you we have a test that's 100% accurate, because there's mistakes that either people sending a shed in mix up a shed or something like that, or on our end we can mix up a shed or make a mistake on entering something. So what we try for is over 99%, which before it was 50% posset or 66, and you have to grow them up and wait till you can breed them to find out for sure. So it's certainly much faster, and yeah, and 99 is certainly better than 50 or 66. But yeah, we won't ever have anything that's 100. But yes, the monsoon test is over 99% accurate. It is a very, very good test. One
Yep.
for sure is incorrect out of like about 350, and maybe two are incorrect. What we've never seen, Charlie or me, we've never had one that we know is het and it's tested negative. That's never happened. So yeah, it's a very, very, very good test, and it does meet the standard we've had from the very beginning, over 99% accurate. That's why we're releasing it now. We had one other individual when we originally we did about 230, 240, and there was one other individual that was clearly not visual, but it tested homozygous. But when we got another shed back from that keeper, it tested negative, and then we had them send us another shed, and it also tested negative, and then we had them send scale clips, and it also tested negative. So yeah, we know that one was just a shed mix up. And so when that happened, especially because we had also run another 100 or 120 that were all correct, that's when it bumped it up over 99%, and that's why we're good to go.
Makes sense. And the thumbnail that I'm showing here is the like I think it's literally just called Monsoon Testing is Here or something along those lines. In our last episode of Reptile Genetics Weekly, we hear it right from Charlie and Ben, like their process. Yeah. So if you want to learn more, definitely watch that episode. It's like 40 minutes. We go into the nitty gritty of what was going on with Monsoon, and I guess what is going on with it.
Yeah, and those are definitely good questions. Silver Serpent says, yeah, just scary when you say it's not the correct marker. It is the correct marker. What I'm saying is it may not be the causal mutation. It may not be the actual mutation that causes monsoon, but there's thousands of molecular or genetic tests out there that test for markers. That's a very extremely common thing. And what we've shown is this marker, even if it isn't the causal mutation, it's so highly correlated with the causal mutation that we see an inconsistency less than 1% of the time. So that's a very, very good genetic test. Is it perfect? No. And even if we know for sure, like with pied, we know for sure as sure as we can be, because we've researchers have induced that mutation in a lizard and gotten a similar phenotype. It's a little different, but it looks like it is indeed the gene and the type of mutation that we were wanting to test for with the pied test. So that's as confident as we can be that we have the correct mutation and that it is the actual causal mutation. You can still have weird problems pop up. You could have a mutation pop up in a line where one of the primers is, or something else can happen that can make the test not work in some individuals, but it's going to be extremely low probability, especially for pied. But even for monsoon, we already know it's going to be less than 1% of the time. So
oh, hey Charlie.
Hey, we're
Long
just
time
talking
no see.
monsoon.
Yeah. There is a question from so Geeked, and I think you just answered his question. Here, I'll pop on here too. He said, for clarity, what you guys said in the video, is it true you were testing for something that isn't actually monsoon? You just mentioned that it is a marker very, very close by rather than the causal mutation, which you're still hunting for. But
that's
what it's pretty much always tied to it. Yeah.
There is a chance it is the causal mutation, but we don't know that for sure. I would guess that it's not the causal mutation, and we will be able to find that eventually. And then that one out of the 350 that's clearly wrong, that one would test correct for the causal mutation. That's what we all think right now, but it could end up that there's something weird with that animal. Snake Morphs one said, could that one be a chimera? Is a chance for homozygous in the clutch? Yeah, I mean,
Ooh.
that's also a possibility. We've shown with the desert, with the animal that was split down the middle and looked
Oh
desert ghost
yeah.
on one side and not on the other. We definitely can take a shed sample from one the DG side, and then one from the non DG side, and the genetic test tested homozygous for one side and het for the other in the same individual. So it's possible there could be a little patch, even if it doesn't look that much different. It could be possible that a patch in it is homozygous
Mhm.
and then the majority of it's not. That's also a possibility. So when you get down to such a small number out of, 350, really just one that we're really concerned about, then there's these really weird, very low probability things that can happen. And so we don't know for sure, but yeah, that's very, very normal. Lots and lots of genetic tests out there don't test directly for the actual causal thing. It's something nearby that's highly correlated. That's very normal. You just need to validate, make sure that it's there the majority of the time, and that's what we've done here.
Fair enough. And again, highlighting geeked, it's like, yeah, hopefully the influx of sheds coming from paid tests will help you find the actual one. That's what I'm hoping for too.
Yeah.
Because either you're going to get like a correct test 99%, 99 plus percent of the time, or you're going to be the person that helps us tease out where the actual causal mutation is, maybe figure out that last shed. So pretty cool deal either way.
Yep.
Um,
And then if that individual gets bred and has offspring, we'll definitely want sheds from that one individual that tested incorrectly, and then we'll
yeah.
have those offspring to also look at. But once again, you have to remember, out of 350, that's the only one that's for sure wrong. So the vast
Mhm.
majority test great.
Yeah.
Then Snake Morphs one also says, or maybe expression blocking conditions, like really a monsoon, but just not expressing. Yeah, I mean, that kind of
Ooh.
stuff can happen. We've had a clown that tested to be a het, is very clearly visual clown that tested to be het, but that's one out of, by this point, tens and tens of thousands. So there's some weird genetic thing going on there. And this maybe we run another 3,000 monsoon tests, and this is still the only one that's weird. That also could happen. We just happen to get this one really weird one in the first 200. That's
Mhm.
why we're very happy to accept over 99%, because it's definitely not perfect. There will be exceptions. It's whether the exceptions are 1 in 100 or 1 in 1,000 or 1 in 10,000 or 1 in 1,000,000. There's going to be weird stuff. They're living and breeding and recombining individuals and DNA, and so there's going to be weird stuff. And
Yeah.
what we've been able to show now is we only expect to see that weird stuff less than 1 in 100. So that's good enough. We're happy to do that as a test.
Yeah. Mr. Tripaz, I wasn't forgetting you, I promise. Was going to ask, I have a geeky genetics question. Have you ever discovered morphs that cannot be combined because they're on the same chromosomes? And have you ever used fish analysis to look at genes? I don't know what fish analysis is, so
So yeah, we
I'm curious
have
too.
actually here in this graphic that Kayla has up, many of
Yeah.
them are part of complexes. And so the yellow belly complex, there's a publication on that. Also um, all the blue-eyed Lucy complex, the eight ball complex, the spider complex. So all of those are close enough to each other, either in the same gene or genes that are very close by, that we do see that. So how we talk about them with ball python breeding is we call them acts like a super. So if you breed a yellow belly to a asphalt, you can get some that are, or if you breed a super, a super yellow belly, homozygous yellow belly to an asphalt, everything's going to get yellow belly. Some of them will get the asphalt, but then when you breed an asphalt yellow belly to a normal, you're just going to get either yellow bellies or asphalts. You're not going to get that combination if it's bred to a normal. So that's kind of what you're talking about there. We haven't done fish with any ball python stuff because it's just kind of difficult to do and would be expensive unless we were already set up to do fish with reptile cells. We could pay a lab that's already set up, but it would be pretty expensive and not something that we've had a need for yet. But that's just a way to use fluorescence to look for if there's two different sequences or mutations that you're interested in, and then you could use fish to find out where they are. But the other reason we don't really have a need for that is we do have a very high quality whole genome for the ball python that we spent a lot of time and money on. And actually this last week, I just got an update in the number of annotated genes and all that kind of stuff. The quality is up even higher on this reference genome. So we already know if we have two different sequences we're interested in, we can look on our reference sequence and we can see if they're right by each other or on different chromosomes or stuff like that. So we don't really have a need for fish because we have that really good high quality whole genome.
Yeah. Oh, girl next door 931 commented that apparently a local near her, Reptile Collective Steve Beamer, had a Zuwadi ball python that you almost got. If you do, yeah, he's awesome. I actually got my I met him at Colubrid Fest, which I think I'm wearing the shirt for it today. I got my scaleless rat snake from him, and he's also sent us a bunch of sheds to help us with like corn snakes, rat snakes, etc. So, and probably ball python stuff too. So that's awesome. Highly recommend. His animals look great.
Yes, and if you do get it, then send the shed in to us, or have him send it in, or both.
Maybe I can shoot him an email, bully him into sending it to us. Respectfully bully, it's fine. Let's see. Very cool. Thanks. I know you said with Monsoon there were 6 locations with similar sequences, which might have been confounding the results. As a follow-up to Mr. Tripaz. I hope I'm pronouncing that right.
Yeah, and I didn't worry about where those were. I could take the time to see where they were, but just seeing that there was that many different places with homologous sequence, I was like, okay, that's why this is such a pain. But yeah, I think that some of them were on other chromosomes. I think some were close, at least on the same chromosome, and then others were somewhere else, but I don't remember for sure. I just was very sad to see. Well, it made sense why we're having such a hard time designing primers that worked well, but yeah, it just means that it's going to be a pain to get that to work. That's for sure.
Yeah. Leviathan. Hey guys. Thanks for tuning in.
Yes.
Let's see. Royal X. If that's Xander over there, I was asking about the Desert Ghost, DGA and DGC. Do we know how they? Any updates on like if any of those enhance color at all? Because I know people are still curious about enhancer.
DGC is the one that causes Desert Ghost, so it definitely makes a difference. DGA and B, as far as we can tell, the person that did the most testing when we were testing for A, B, and C was Justin at Canova, and they said they didn't see any correlation with A or B, like being the more silver ones and the more yellow ones, or any of that kind of stuff. So not anything that we can tell.
Yeah, I definitely trust Justin's eye on that. I feel like if anybody could see it, he might be one who would.
And the A and B could also just be improving the overall health of the individual instead of making it look better. Maybe that helps them have a higher probability of breeding and stuff like that. That's possible. We're still not sure why there is that correlation, but now that we can test for it with just one spot, we're less worried about that.
Fair. Hi, Heart of the Python. Thanks for joining. And Margaret from Puerto Rico. Hey.
Yes.
Thanks for joining.
Awesome.
Yeah. Let's see. I know it's about 5 minutes until Ben's got to go today. He's in Utah right now and is going to be traveling very early in the morning. So, get in any last minute questions, whether it's monsoon or totally different. Hi, Silver Scale. You just got here. I'm sorry. Next week we'll have most likely have a proper hour long stream. It's just been a crazy week with travel for Ben. So.
Yes, and on IG, Snake Morse One says any plans for DGB fast test? No, I don't think so. There's just not really that much interest. Like we said, it may be providing an improvement, and that's why it was highly correlated. But that one, when we're talking about the monsoon one, that was so highly correlated. We're above 99%. With DGB, it was lower than that. It was more like, I think it was like
60 or
93. No, it was pretty good. Yeah, it was pretty good, but it wasn't over 99. So, but yeah, I don't think we'll do a fast test for it. It would be something you'd need to request, just because there's not that much interest in it now that we don't see it. If it also was more likely to have the more silver looking or the more yellow, or like if there's something we could see, then we would be more likely to make a test out of that. But I would say what we're getting back from breeders is they don't see that correlation. So
fair enough. From Brew Balls, any updates for tri-stripe and paint tests?
Not yet. No, still working on them.
Fair enough. I know on this graphic, tri-stripe wasn't on this one. Are you good on sheds for tri-stripe? Or should I add it to the sheds wanted thing?
We have a pretty good amount. If you have homozygous tri-stripes, the visual tri-stripes, we could still use a few more. I probably would never turn those down until we actually have a tri-stripe test, because now that we've found out from many different mutations we're working with, it's very common we end up having a secondary line that we also have to design and test for. So
RK asphalt.
So yeah. Yep. RK asphalt. That's another one that's like that looks very similar to the asphalt we already have, but it doesn't test the same. So.
So Geek is asking as a follow-up to the Desert Ghost conversation. So are there DG visuals that only test positive for DGC and no DGA or DGB? I'm curious about that too.
I don't think we've ever seen that where it's negative for both. I can't remember any completely negative for both A and B. Right. I think it's always at least one or the other or both. But there are definitely times where it's visually a Desert Ghost tests homozygous for DGC, and it's either negative for A or negative for B. That does happen, but yeah, It's not as often.
Fair enough. I feel like it was never officially stated in a clear way, so I never truly understood what happened there. I feel like, sorry, Ben, go ahead.
Yeah, I mean, we've talked about it many times in different places. It seems like people hear little pieces or don't, they miss whatever. So we've tried. We will. So Steven from Leviathan, hey, once again, if they're still on here, him or Courtney's still on. But we did record a documentary episode with him where we talk about that in fairly good detail.
Yeah.
So hopefully that will get enough views from a broad audience that will help, to make sense. But yeah, that's, in a nutshell, people really wanted a DG test. We had something that worked pretty well. We called it an elimination test. It was listed as that in Morph Market and in Clutch. So it was different from the other tests. It lets you know, hey, if you get a negative, it's most likely correct. If you don't, that's our best guess. It was right. I think it was either 93 or 95%, but it wasn't up to where we, want it to be. And that's why, because of Desert Ghost, that's why with Monsoon, we waited so long to make sure we could get it above 99% before we released it. So,
yeah, because it seemed like as soon as,
once the DGC came along, we knew that was the correct one, and that validated over 99% pretty quick. And then we didn't need A and B anymore to be able to do DG testing. So,
yeah. Let's see. So do you still need DGA or DGB for the DGC to present? Like, are they epistatic?
No, as far as we can tell, it's just like clown or pied. It's just DGC that we test for. That's all you need to worry about.
Mm-hmm. Simple recessive. So it's more likely that DGA and DGB are just something that travels along with it, that were our first really close hits. And well, people latched onto that. And it was really exciting at the time, but genetics are very good at giving you something one way at first, and then next you're like, oh, actually it's something completely different. Which again, that's why we held off on Monsoon for so long.
We were getting pressure for DG every single day. People really wanted
Oh, yeah.
it. Like, if it's 85% correct, I'd still rather have that than 66 or 50. But then if we release it, there's enough people, the broad, group of people that aren't spending the time, watching videos and reading and asking questions. So they just assume if they get a test and it says something, then that should for sure be right. And so, we learned through that DG fiasco that we can't release something, even if we try to list it different and try to let people know we're looking for something more. And we did find something more and it made it much better. But, people latched onto that before we had the DGC part of it and ignored the fact that we gave as much warning as we could. So yeah, we knew with Monsoon that we had to keep testing and waiting until we got above that 99%. So we learned, we just had to tell people over and over again, sorry, Monsoon's not ready. Sorry, Monsoon's not ready.
Yeah. It does seem odd that you haven't seen any without either. That doesn't make, oh,
sorry.
Oh, you're good. Just with how many tests we've done, fair. But I think, to be fair, we're mostly just testing for C now, and it's I don't think that we're also documenting A and B.
Well, and A and B are highly correlated, so I mean, it's not going to be very often that we don't get it. The way that Travis explained it, I liked. It's like if there's a massive earthquake and you and your partner are sitting on a couch next to each other, it's very unlikely there's going to be a giant crevasse form between you and your partner. But if you and your partner are in different cities in the same state, it could happen. It's not that likely, but it could happen. But it's more likely than if you're sitting on a couch next to each other. But if you're in different states, then the chance that a big crevasse would occur, jumps up quite a bit. So A and B were probably like, in the same city, but not in the same house or, right next to each other.
Yeah.
Now the one that we have is either the causal one or they're sitting on the same couch. Like we don't see that problem. So that's when you're thinking about recombining and things traveling together. I think that when Travis said that, that's a good way to think about it. If you're two different, the actual mutation and the one that you're testing are super close to each other, the chance that there's going to be a crossover event in between is extremely low, and that correlation is going to be super high, and it's going to be a great test.
Yes, what TripAd says, chromosome recombination rate. Yeah, when they pretty much.
Yeah, and then the question about sex-linked morphs. Yes, we actually did a big episode. I think it was 2 episodes ago on banana and coral glow in ball python. That's definitely a sex-linked trait. And so if you haven't read up about that, in that episode, we give a link to a scientific paper that also was published about banana in ball pythons and how that linkage works, and the fact that there is recombination going on in the sex chromosomes, which is different for humans and most species where we think about sex-linked traits. In reptiles, at least in ball pythons, there actually is recombination going on, a fairly large portion of the sex chromosome.
That's so cool.
That trait, if it's on an X chromosome and it's a male, so he would be passing it to his daughters, it will pass like 96, 97% of the time. It will only go to daughters. But then 3% of the time there will be a crossover between the sex determining region and the actual banana mutation, and that male making or that female making sire will actually produce a male offspring that's a banana, but that's only like 3% of the time. So yeah, Mr. Tripaz, yeah, you'll have to check that out if you haven't seen that yet. It's pretty interesting, and took us a little while because a lot of us that are genetics geeks aren't used to thinking about a sex chromosome where a large portion of it recombines. So it was kind of a fun problem to solve.
Yeah. And Deathhorn Dist, is there any work on what causes the extra darkness to happen in TSK?
Yeah, we're working on that. We have samples. No answer yet, but yes, we are definitely working on that, and we could for sure use more samples if you have, the really extra dark animals they seem to be from the TSK line, or the black axanthic, which we know now is the same mutation. The main mutation that causes it is the same, but there is likely a secondary mutation that causes these super dark ones. So.
Awesome. Cuddle Time Critters got black. Oh, MBKs. Oh, cool. Congrats on your eggs.
Yes. Eggs. This is the best time of the year, that's for sure.
It is. Second only maybe to when they start hatching. Hi, Spirit. All right, Ben, it is about 8 minutes past your go time. So do you want to call it here, or what you want to do? Or do you have wiggle room?
I probably better get going. I'm just seeing if there's any. Looks like there's not any more.
More questions on Instagram?
Yeah, let's see. Let's do one more. Snake Morse one. He says, hard to gather stats on if DGB has any benefit. It's hard to test, so most breeders don't know their DGB status. Yeah. So especially because we're not really running that anymore. But with the amount of DGB tests that we did, there just didn't seem to be a correlation. So that's why it's kind of getting dropped.
Fair enough.
But if you keep tracking it, Randy, Snake Morse one, let us know if you feel like you do see something. We could certainly bring it back. So
yeah. Cool.
All righty.
All right. Well, thanks everybody for joining. I think there's, make sure I got everything. Yeah. All right. So everybody be sure to like and comment, subscribe if you haven't already. Thanks for joining our live stream. It was a shorter one today, just because Ben's traveling, but we appreciate you tuning in. And next week we'll have a full live stream, hopefully with a guest with us too. So
yes, and I will be less run down. I'm definitely tired from traveling, so I'll have more life in me.
Fair enough. You do look like a father of four who's been traveling across the country all week, so yes. All right. Thanks everybody, and I guess we'll see you next week.